Hello, biology. The purpose of this video is to allow those who missed any of the 2-4 notes to get them caught up, or maybe students that I went too fast for to get caught up. But without further ado, here we go. Remember, all class notes need to be in Cornell note style. If you are typing these, you're going to need 1, 2, 3, 4, 5, 6, 7, 8 rows, 2 columns. If you're handwriting, draw a line down the center of your page. Remember, number them Cornell note style. Questions on the left, answers on the right, starting with number 30 and ending with number 37. All right, here we go. If you're turning down the volume so you can listen to your music, or if you just simply don't want to hear me, the way that you will know I am about to hop ahead to the next slide is I will do this. I'll try to give you about a minute and a half per slide to write things down. But of course, this is a video, so you can pause the video anytime you want. Game day here at the Paulus Pad. Dolphins and Seahawks. Not looking so good so far, but go Fins. And of course, this jersey is from one of my favorite players from back in the day, a walk-on, Zach Thomas, and Hall of Famer. If not for the league, definitely for the Dolphins. I think he's going to be a Hall of Famer, or if not, he might already be a Hall of Famer. I should probably know that. All right, here we go. Moving on. All right, so RXN is going to stand for reaction. These are processes that change one set of chemicals into another. Some are really slow. Iron turning into rust. Remember, iron doesn't actually need water to turn into rust. Just needs to be exposed to the atmosphere. It's the oxygen in the atmosphere that turns it into rust. Water speeds it up. And of course, hydrogen gas is highly explosive. All right, parts of a reaction. All right, so the reactants are what are on the left side of the arrow. Reactants react. They go into a chemical reactions, and bonds are broken, and it takes energy to break and to make bonds. And then the product, these are things that come out of a reaction. They're elements or compounds or molecules that come out of a reaction. They're on the right side of the reaction arrow. And this is when bonds are being formed. Perfect example, hydrogen ion plus hydroxide ion. Remember, hydrogen ions are positive, hydroxide ions are negative. Those would both be reactants. And then you're going to have an arrow going to the right, and then you're going to get water. Those two ions form an ionic bond to form water. That would be the product. Okay, I'm getting ready to hop ahead to the next slide in about 15 seconds. And here we go. All right, so energy, we're going to use a capital E. Remember, RXN is reaction. So energy is either released or absorbed when bonds break or are formed. It does take energy to break and form bonds. Chemical reactions that release energy occur spontaneously, and those that absorb energy won't occur without a source of energy. And every organism must have energy for chemical reactions. Plants get their energy from chemical energy that they get from glucose, that they get from photosynthesis. Animals get their energy from eating plants or eating things that eat plants. So we have to get our chemical energy from the food molecules, carbohydrates, lipids, proteins, to a small degree, nucleic acids, because typically like live produce does have living cells in it and has nucleic acids in it. But typically we do not consider nucleic acids as a nutrient or a foodstuff. And remember, plants don't get their energy from the sun. They have to convert carbon dioxide from the atmosphere, water vapor from the atmosphere, into glucose. That's where the plants get it. The sun enables that reaction to take place. The photons that are in solar radiation, is what actually runs that reaction. Okay, getting ready to bump ahead to number 33 in about 10 seconds. All right, here we go. Okay, so enzymes, pardon me, are proteins. They're special proteins that help speed up reactions. So important reactions that take place in the body where the products are really necessary or the reactants just simply need to be broken down. For example, hydrogen peroxide. We're going to be doing a lab on that. It depends on when you're watching this video. This week, I'll be showing you some videos of that lab. Hydrogen peroxide gets toxic when it builds up in cells. So catalase enzyme speeds up that reaction to break down the hydrogen peroxide so the cells don't become toxic. Not all enzymes are catalysts, but all catalysts are enzymes. Well, all organic catalysts are enzymes. Most enzymes, though, are catalysts. And a catalyst is just something that speeds something up. Enzymes are very specific. Usually their shape only enables them to catalyze. Catalyze means to make happen. Only one chemical reaction. So reactants or substrates, as you'll learn in a little bit, need to collide with energy. And that energy is called activation energy. And as soon as there's enough energy to make that reaction take place, then the reaction happens. That energy is necessary to break the bonds of the reactants or the substrates and to make the bonds of the products. Getting ready to hop ahead in about 20 seconds. All right, and here we go. So you do not need to write down these questions. Some of you guys do, because I do take some of these questions and put them on tests. These are just for you to practice with the content. Chemical reactions are blanks that change one set of blanks into another set of blanks. Take a look back at number 30 if you're not sure what the answer is. And of course, if you're looking at number 30, chemical reactions are processes that change one set of chemicals into another set of chemicals. All right, next one. Remember, on these, you need to choose, does this go in the first blank and this in the second blank, or does this go in the first blank and this in the second blank? That's what it means when there is a dash in between them. Blanks enter into a chemical reaction and are on the left side of the reaction arrow, while blanks come out of a chemical reaction and are on the right side of the reaction arrow. And if you're not sure, you might want to take a look at number 31. Reactants enter into chemical reaction while products come out of the chemical reaction. Enzymes that speed up the rates of reactions, what are those things called? You might want to take a look at number 33. Enzymes that speed up reactions are called catalysts. All right, here we go. All right, so the enzyme substrate complex. Remember earlier I said that reactants, if we're talking about an enzyme reaction, are called substrates. Enzymes provide a location where the reactants come together to react, and that reduces the activation energy needed for reaction, which allows them to speed up. If you need less energy to make reactions take place, then the reactions are going to take place faster with the same amount of energy. The reactants of enzyme-catalyzed reactions are called substrates. So, for example, in the catalase, in the enzyme activity lab, because we're not just working with catalase, hydrogen peroxide. In both cases, when we're doing manganese dioxide, which is an inorganic, non-living, it's naturally found in nature, catalyst, and catalase enzyme, which is an organic catalyst, those both break down hydrogen peroxide. So hydrogen peroxide is the substrate or the reactant, and oxygen and water, two harmless substances, come out of that reaction. Those are products. And the reason why it's important to break down hydrogen peroxide in our cells is because when it gets concentrated, when there's a lot of it, it becomes toxic, and it can actually do damage to the cell and the things inside of the cell. The fit between enzymes and their substrates are so precise that we usually refer to them as a lock and key. They have three-dimensional shapes, which is like a lock and a key, that only enable one enzyme to bond with one substrate. And if you remember back to when we were talking about proteins and amino acids, the monomers of proteins bonding together, depending on the amino acid, which again really depends on the side chain or the R group, gives them different folding methods and different sheeting methods. And depending on what amino acids are in this polypeptide, this long macromolecule of amino acids bonded together, is going to determine the shape. And of course, the shape, if it's an enzyme, determines the substrate and what reaction is catalyzed. All right, that did not mean I'm going ahead to the next slide, but we're about to go ahead to the next slide, on to number 36 in about 10 seconds. 5, 4, 3, 2, 1. Okay, so what happens after the enzyme substrate complex is formed? So what happens after the bonds between the substrates and the enzyme are broken? The enzyme and the substrates remain bound until the reaction is done, and the substrates are converted to products. So it stays there until the bonds are broken, the bonds are reformed to make whatever product. So if we're talking about hydrogen peroxide, we're going to bond two oxygen atoms together, O2, and then we're going to bond two water compounds together. So two hydrogens, no, one water, if I'm not mistaken, one water. And I'm sorry if the equation is not balanced, but you only get one extra oxygen from the hydrogen peroxide. So two reactions would need to take place for those two oxygens to be available, and then they make a bond to form an oxygen. Okay. When you get to chemistry, you'll know what I'm talking about, balancing equations. All right, the products of the reaction are released, and the enzyme is free to start the process again. So after the products leave, the enzyme is still there, waiting for another substrate, waiting for more energy to make the next one happen. It would make no sense at all for the body to destroy the enzyme after one reaction. The enzyme just sits there, and when substrates bump into it and it's got activation energy, it makes it happen again, and then again, and then again, and then again. Now, for those of you out there that are meat eaters, you probably know that there's a lot of protein in meat. There are a lot of enzymes in meat, okay? But we usually don't eat raw meat. We usually cook it. And the reason why we cook it is it makes the texture and the taste better. So whenever you change the pH, the acidity or alkalinity, or the temperature of the environment that enzyme or protein is in, you have the opportunity to do what is called denature, to denature, denature, or denaturization, which basically means breaking bonds that make up the enzyme, breaking bonds that make up the catalyst. So you destroy the ability for that enzyme to operate. And that's one of the reasons why buffers, if you remember back to section 2-2, are really important, is because they maintain the pH. So if you have reactions that are taking place at a certain pH, those buffers make sure the pH stays correct and it doesn't allow damage to be done to the enzyme, so the reactions, important reactions, can continue. All right, so enzymes work best at certain pH values, and many are affected by changes in temperature. And in the catalase lab, or the enzyme activity lab, I will be boiling liver, and then we will compare the reaction speed to a chunk of liver that has been boiled to the reaction speed of a chunk of liver that has not been boiled. And you'll notice in that reaction that very few, if any, bubbles, evidence of oxygen, very few bubbles come from the boiled liver, meaning that those enzymes have been destroyed, their shapes have been modified, and those reactions can no longer take place. All right, getting ready to bump ahead. This is the last item of the notes that you'll need to be writing down. Although I would encourage you to stick around and watch a little bit more of the video, as we're about to do the whiteboard questions that we don't use whiteboards for. What's another term used for a reactant that is involved in an enzyme catalyzed reaction? Real careful. What's another word for the reactant? And if you're not sure, you might want to take a look at the second bullet for number 35. Yeah, substrates are referred to as reactants when we're talking about an enzyme catalyzed reaction. That's a mouthful, by the way. All right, the enzyme substrate complex stays together until substrates are converted into, so that substrate and the enzyme and their three-dimensional shape stays together, bonded, until substrates get converted into what comes out of a reaction? Products. Changes in blank and blank can affect how well enzymes catalyze chemical reactions, changing in pH and temperature, or changing in the shape and number. And if you're not sure, you might want to take a look at number 37. Yeah, it's changes in pH and temperature affect catalyzed reactions. Okay, I wouldn't necessarily recommend writing these down, but some of you are, because some of these will be on the test. But these are typically questions that you're going to hit in your reading notes, you're going to hit in your class notes, we would have hit on assignments, so I don't know if it's necessary to write them down, but some of you will, and that's not a problem to do things just in case. The elements or compounds produced by a chemical reaction are called, if you're not sure, take a look back at number 31. Remember, reactants react and products are produced. Chemical reactions always involve changes in energy, enzymes, catalysts, changes in the atomic number of the reactants. Wow, that sounds really intelligent. D sounds really good, right? No, chemical reactions always involve changes in energy. Activation energy to make it happen, spending energy to break bonds and make bonds. It's changing the amount of energy. The factor that prevents many energy-releasing reactions from occurring at relatively low temperatures, what do we call that? Catalytic energy, chemical bonding energy, enzyme energy, or activation energy. And if you're not sure, you might want to take a look back at number 32. Yeah, you're right. That energy that is necessary to make reactions take place is called the activation energy. Which of the following statements is true? All proteins are enzymes, all catalysts are enzymes, all enzymes are catalysts, or all catalysts are proteins. A couple up there are partially true. There's only one that is 100% true. All enzymes are catalysts. All right, what happens to an enzyme after the reaction it catalyzes has taken place? And I'm sorry that there's some stuff blocked there, you guys. The enzyme is destroyed and the cell must make another one. The enzyme holds on to the product until another enzyme takes it away. The enzyme is unchanged and ready to accept substrate molecules. The enzyme changes shape so it can bond to a different type of substrate. It doesn't make any sense for it to change shape because it's a lot of energy, cell energy, has been put into making that shape right, so it's not D. Why hold on to the product until another enzyme removes it? That means you need two enzymes for one chemical reaction. That doesn't make a lot of sense. Destroying the enzyme, are you kidding me? It takes a lot of cell energy to make these enzymes, make these proteins. The one we're left with is C. It is unchanged and ready to accept another substrate molecule. All right, ladies and gentlemen, that is it for the notes. Don't forget to post these on the classroom. Make sure that they are numbered. Make sure that they are in Cornell note style. And we'll see you later, and go Dolphins.